Welcome to Microbiology Lab. In today's lab session, you'll be learning about determination of antibiotic sensitivity assay by Kirby-Bauer assay. So the aim of this experiment is to determine the sensitivity of bacterial isolates to antibiotic using the Kirby-Bauer assay method, that is a agar disc diffusion technique. So the principle behind this technique is antibiotics are antimicrobial chemical compounds produced by microorganisms to combat other microorganisms. So these compounds can be used to treat microbial infections in human. This agar Kirby-Bauer disc diffusion susceptibility test, it aims to determine the sensitivity or resistance of a pathogenic aerobic or facultative anaerobic bacteria such as E. coli, Salmonella, Shigella, Pseudomonas, or Klebsiella, Stenotropomonas, Clostridium. So facultative anaerobic bacteria to various antimicrobial compounds. Usually, pathogenic organisms are grown on Muller-Hinton agar in the presence of various antimicrobial impregnated filter paper disc. So here in this method, the presence or absence of growth around the disc is an indirect measure of the ability of that compound to inhibit the microorganism. During the incubation, the antibiotic diffuses from the disc into the agar with decreasing amounts the farthest it is from the disc. If the organism is killed or inhibited by the concentration of the antibiotic, there will be no growth in intermediate area around the disc, which is called as zone of inhibition. So this is the expected result. So here the Petri plate, which contains the Muller-Hinton agar, which is lawned by a pathogenic microorganism, and then the antibiotic disc, different discs are kept, such as penicillin, streptomycin, chloramphenicol, ampicillin, like the different antibiotic disc kept on the surface of the agar, which is already coated by the pathogenic microorganism. And it is incubated for 24 hours. And after 24 hours, you can see the zone of inhibition. So the zone of inhibition around the antibiotic disc indicating sensitivity. So here you can see the zone of inhibition. And here there is no zone of inhibition. So this is usually measured using a scale. Here you can see how the measurement is done using the scale. Here it is measured and the diameter is about 20 millimeter, and this is referred to as zone of inhibition. So this is the expected result of this experiment. So the interpretation of this experiment, there can be three types of result as we have seen in the earlier slide. That is sensitive, intermediate, or resistant based on the size of zone of inhibition. So here, if the observed zone of inhibition is greater than or equal to the size of standard zone, the microorganism, it is considered to be sensitive to the antibiotic. So for the patient, the normal dose of particular antibiotic is effective. The second case is if the observed zone of inhibition, if it is smaller than the standard size, then it is intermediate. So higher dose of antibiotic is required for the patient. If there is no zone of inhibition, the microorganism is considered to be resistant. No acceptable dose of antibiotic is effective for that particular microorganism or to that person who is affected by that microorganism. So the antibiotic sensitivity test, it is used to identify the organism based on its sensitivity to various antibiotics. So this is the ultimate aim of this experiment. And now you can watch the video and you can understand how the Kirby-Bauer assay method, it is performed in the lab using the antibiotic disc and then by keeping the antibiotic disc on the surface of the agar lawned with pathogenic microorganism. Now you can watch the video.